welcome back to Bio 111. This is Chapter 14. This is Section 4, DNA replication in prokaryotes. Alright, so we're looking at a model of DNA replication in prokaryotes. Now, I want you to remember, in prokaryotes, the DNA are circular, meaning that the two ends of the DNA strand are connected as a loop, so they're rings. And so what this means is that essentially during replication, you just pull apart the two strands. So if you look at this picture here, you notice that there at the far right we see our double helix just in front of that green ring that we see. It's called topoisomerase. That is the original DNA. And as we start looking to the left, we see the mechanisms of it being duplicated. If we go all the way to the far left, we see two new strands of DNA. Now this topoisomerase, what it does is it untwists the DNA. It makes it straight, linearizes it. Then we have helicase, which breaks those hydrogen bonds between the two sides of the DNA, the two strands of DNA. And the process of replication is taken on by various polymerases, DNA polymerases. All right. Now, in the case of prokaryotes, because it is a loop, there is really no end to it. They just replicate until they meet at the other side. All right. So a replication fork is formed when helicase separates the DNA strands at the origin of replication. Topoisomerase prevents the overwinding of DNA double helix ahead of the replication fork as the DNA is opened up. Single-strand binding proteins bind to single-stranded DNA to prevent the helix from reforming, so they don't want it to bind to itself again. Primase synthesis. Primase synthesizes an RNA primer. DNA polymerase 3 uses this primer to synthesize the daughter DNA strand. DNA polymerase 1 replaces RNA primer with DNA. And then DNA ligase seals the gaps between these Okazaki fragments. These are these short fragments of DNA that are formed as a result of the direction the DNA has to be synthesized in. Right? It's just a result of the geometry of DNA replication. Looking at some examples of some enzymes and their functions. Now, I want you to remember, these enzymes, they're proteins. They're made up of amino acids. We're just kind of connecting things back to previous chapters. They're the machines that do the work of the cell. All right, these are just some examples that are used in DNA replication. All right, we're going to just go right into, because these are short sections, go right into section 5 here. We're looking at DNA replication in eukaryotes. So what happens in us, what happens in animals, what happens in plants, what happens in fungi. It's a little bit more complex. Remember that prokaryotes are smaller. Eukaryotes have much larger DNA. In prokaryotes, you have a single origin replication. You have this single circular chromosome that begins to basically split in half. In eukaryotes, we have multiple chromosomes, and so we have multiple origins of replication. In fact, multiple origins of replication in the same chromosome. And the reason for this is because the DNA is so long, there's so much DNA, it can't be done in just one part. It gets copied in multiple locations, or begins in multiple locations. All right, so we look at the rate of replication in prokaryotes. It's 1,000 nucleotides per second. All right, compared to eukaryotes, which is 50 to 100 nucleotides per second. All right, so think about this. If you're typing, right, if you're typing on the computer or your cell phone, and you're typing really fast, you're going to make more mistakes than if you were typing really slow. And so prokaryotes, they evolve faster, right? Um, Bacteria, you've probably heard of, being afraid of the next superbug because we use too many um, antibodies or because we wash our hands too much or something. The reason why that might be a threat is because bacteria, they evolve fast, partially because they're kind of sloppy at copying their DNA. They make mistakes. Those mistakes increase variation in their genotypes, which in turn gives them something to select against, which is needed if you're going to have evolution. Eukaryotes, much, much, much slower. Eukaryotes are extremely, extremely finicky with their DNA. Eukaryotes do not survive well if they have any sort of mutation. This is where we have many disorders and cancers. All right. Telomerase is not present in prokaryotes. It is present in eukaryotes. We'll see what telomerase is a little bit later. RNA primer removal, two different types here. Strand elongation, we see that that is done by different enzymes in the two types. And sliding clamp, also two different systems. All right, so we talked about telomerase being present in eukaryotes but not in prokaryotes. Telomerase is a enzyme that solves a problem that eukaryotes have, that you and I, plants, any animals, fungi have, and that is because we have a linear chromosome. We have one side that can't be extended all the way, that can't be replicated, right? This is part of that issue we talked about earlier, the Okazawa fragments, where they the DNA essentially doesn't have a way to start because it's at the end of the DNA. And when that happens, the part that is not replicated essentially just stops being replicated in future generations. Now, the way that eukaryotes have solved this problem is by adding a bunch of useless DNA to the end of their chromosomes. We call those telomeres. This useless DNA can be sacrificed in each DNA replication event. Now, this is fine for things like your nerve cells or any other cell in your body that isn't going to replicate too much, right? Because they're not going to run out of this sacrificial stretch of DNA. But in things, say, like your skin cells that replicate constantly, the telomeres may very well replicate, may very well be lost completely. And that's a problem, right? Because then once the telomeres are lost, then you start eating up useful DNA that actually codes for something, codes for a protein. Telomere degradation is associated with aging. And the idea of being able to activate telomerase, which is active in some plants, some really old plants like 4,000-year-old redwoods, and that is also present in fetuses. Now, if we are able to turn on telomerase, we might be able to extend our lives, right? Because some of the aging we see is a result of these degraded telomeres. Now, we talked about in eukaryotes there being issues with mistakes being made. Eukaryotes do not like mistakes in their DNA. And so eukaryotes repair these mistakes. And so mistakes during replication are detected and repaired by DNA polymerase proofreading activity. So it goes back and actually proofreads the DNA and corrects any errors made during replication. This is important because mutations, a lot of times mutations, sometimes they don't have any effect, but sometimes they have effects that are essentially like, causes cancer, right? Right, so there's several mistakes made. Mismatch repair. So in mismatch repair, the incorrectly added base is detected after replication. The mismatch repair proteins detect the base and remove it from the newly synthesized strand by nuclease action. The gap is now filled with a correctly paired base. So mismatch would be where, let's say we have a guanine and a thymine connect. Now, if you look at it, you notice that they're bulged out. This G and T that are highlighted in red are bulged out. That's because they're both large molecules. It causes the bulge out. They're not supposed to be bound together. All right, it gets repaired. The T is replaced with a C, or the G is replaced with an A, whatever is needed to make it work. It would have to, mind you, you would have to use the original template DNA to determine which one you're going to repair. You wanted to keep the template and get rid of the daughter strands. Nucleotide. All right, nucleotide excision. So nucleotide excision repairs thymine dimers. That's where you have two thymines side by side. They kind of get stuck together, and it creates a bulge. You have an extra thymine there. So when exposed to UV, thymines lying adjacent to each other can form covalently linked thymine dimers. In normal cells, these mutations are excised and replaced. So uncorrected replication errors result in mutations. Replication is not a perfect process. Errors occur, and if uncorrected, produce mutants. Mutations are changes in the nucleotide sequence of DNA strands, and they can lead in changes in protein sequence encoded in the DNA. Now, sometimes these changes are beneficial, right? But many times they're not. So there are several types of mutations. Point mutations, they're silent, missense, and nonsense. So silent, that has no effect on the protein sequence. So essentially this happens when you are, there is a change in the amino acid sequence, or sorry, there's a change in the nucleotide sequence, but it actually doesn't affect the amino acid sequence that's generated by that one. So it has no real effect. Missense results in amino acid substitution, so it actually changes the way that the DNA is transcribed, and then the RNA is translated into a protein. Frame shift mutations, this is where you insert an extra nucleotide, or you delete one, and this causes what's known as the reading frame to change. And the reading frame is important. Think about it this way. Let's say that you had a book and you removed all the spaces from all the words in the book. You would have to determine where one word ended and one word began. And that's effectively what DNA is. All the words are joined together with no spaces. If you remove one letter, then it shifts the reading frame. And that can produce a different sequence of amino acids. All right. There are also chromosome mutations, insertions, deletions, translocations, and inversions, so flipping the DNA around inside the actual chromosome, fusions and duplications. All right, and that is it for chapter 14.